Welcome to today's QAAFI Science Seminar. Could I have the next slide, please? I'm Robert Henry, Professor of Innovation in Agriculture in QAAFI, and I'll be facilitating today's seminar. Could I have the next slide, please? I'd like to start by acknowledgement of country. I acknowledge the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Can I have the next slide, please? Today's seminar will go from 12 till 1. We'll have a question section at the end, and we'll ask you to type your questions into the Q&A tab rather than the chat function. Next slide, please. So today's seminar is by Upuli Nakandala. Upuli has come to us from Sri Lanka and has joined the Horticultural Tree Genomics project in QAAFI. And today she's going to talk to us about citrus genomics. So, Upuli, can you take it from there?
Okay, thank you very much. First of all, thank you very much, Robert, for introducing me. And also, I should thank Robert and Craig both for giving me this opportunity to present my work in this seminar. So today my presentation is about a chromosome-level genome assembly and annotation of Australian citrus. Okay. So before going into details, first I would like to give you a brief introduction about the citrus growing regions in Australia. There are two major citrus growing regions in Australia. One is in Southeast, which includes Riverina, Murray Valley, and Riverland regions. And the other is in Southeast Queensland, which include extensive growing regions of Central Burnett. In addition, there are minor citrus growing regions also exist in New South Wales, Queensland, Northern Territory, and Western Australia. Navel oranges and Valencias are the main popular orange varieties, and Imperial is the main Mandarin variety, which are grown in Australia. In addition to them, grapefruits, limes, and lemons are also grown in different regions in Australia. Australia exports fresh oranges to over 35 nations, with the large export destinations being in Asia. If we talk about the native citrus production, there are 6 species which are endemic to Australia, out of which one species, Citrus gracilis, is endemic to Northern Territory, while the other 5 species are primarily found in Queensland, while some of them are also found in New South Wales and South Australia. So in this image, on the left side, we have all the wild citrus species. So as I mentioned earlier, we have 6 species: finger lime, desert lime, round lime, Russell River lime, Mount White lime, and Kakadu lime. And on the right side, we have a selected set of domesticated cultivars, which are or have been important in Australian citrus industry, which are Murcott Mandarin, IRM2, Allendale Mandarin, and Premier. So let me give you a brief introduction about the citrus genome. The citrus has a basic chromosome number, which is 9. So in diploid species, we have 80 number of chromosomes. So diploidy is the general rule in citrus, but we also have some triploid and tetraploid varieties as well. For example, triploid Tahiti lime and tetraploid Poinsettia trifoliata and wild Hong Kong kumquat varieties. So citrus species have relatively small genomes, usually range from between 340 megabases to 380 megabases. And the genomes of some of these species have been sequenced and assembled over the past few years. So the very first genome that are available is for sweet orange, which was published in 2013, and one for clementine, which was published in 2014. Other than that, we also have some other genomes available for the other species, as you can see here in this slide. So we know that the high quality reference genome is the key resource for plant breeding, which provide more accurate predictions of genes in plants, and also to better understand the genetic underpinning of complex agronomical and physiological traits in plants. So traditional breeding methods have been successfully used to improve the citrus cultivars and also to develop the new varieties in citrus. But this has been done with limitations and difficulties, mainly because citrus trees are very large and also they have a large juvenile phase. And in addition, citrus plants also have complex reproductive biological characteristics such as parthenocopy, polyembryony, self-incompatibility, and heterozygosity, and so on. Because of those reasons, using traditional breeding methods to get the desired traits of plants during a short period of time is not that easy. And also, traditional breeding methods are restricted to actually get the, to get the traits like related to fruit quality mainly, such as a number of seeds and fruit ripening time, and so on. So because of that, in that context, genome-assisted breeding is very helpful, where the availability of high-quality genome can provide major sources of valuable genes to get the improved varieties through molecular breeding approaches. So far, we have a few citrus genomes which have been anchored at chromosome level, and some of them are also haplotype phased. So the accuracy and the completeness of these genomes vary in different levels. So the most recently published citrus genomes are lemons and for sweet orange. So we have two lemon genomes. One is published in 2021 and the other one is in 2023. So we have a good reference genome for sweet orange, which was published in 2023. And also we have another good genome for Poncilis trifoliata, which is a close relative of citrus, which was published in 2020. But so far, no reference genomes are available for Australian citrus. So the main objective of our project is to develop a good high quality genomes for Australian citrus. Yep. So before going into detail, let me briefly explain about the genome assembly. So genome assembly, by definition, is the process of taking a large number of sequencing reads and put them back together to create a representation of the original chromosomes from which the DNA was originated. So then what's the de novo genome assembly? So de novo means we have no prior knowledge about the genome sequence or genome layout or the genome composition. So basically, we do not know anything about how the genome looks like. So in this process, first we start with the genomic DNA of the target organism, and then it is broken up into million number of pieces, then read on a sequencing machine, creating sequencing reads. The sequencing reads can be either short reads or maybe long reads. So their reads, the lengths can be varied. And then these sequencing reads are assembled to form contigs. So here a contig is a continuous piece of sequence containing no ambiguous nucleotides or no unknown sequence, which is denoted as N. And then these contigs are ordered and oriented to generate scaffolds. And finally, these scaffolds are joined to form a chromosome. So first we started the path by smart sequencing to generate hi-fi reads, and the smart sequencing was performed at AGRF University of Queensland. So we produce the hi-fi reads. So these hi-fi reads are high fidelity reads. They are usually having like long lengths, which is usually range from 10 to 25 kilobases. And they have a high accuracy, which is around 99.9%. So first we started with high molecular weight double-stranded genomic DNA, and then they are fragmented. And then we go for library preparation, as you can see here on this left side image. So in the library preparation, this high molecular weight double-stranded genomic DNA, fragmented DNA, is kept on both sides with ligated hairpin adapters where the sequencing primers can attach. And this creates a circular template for the polymerase to navigate. And then the sequencing is performed using PacBio SQL2 system. So as the polymerase moves through this molecule again and again, it generates a number of subreads, finally can be consolidated to a consensus hi-fi read. So the more number of passes here we have, the higher the accuracy of the final consensus read. So that is about PacBio Smart Sequencing. And in addition to the hi-fi reads, we also have another type of read, which is known as Hi-C read. So the library preparation and the sequencing was performed by Ramashetty by Face Genomics. So here in this process, what happens is, first we harvest the fresh leaf materials, and then the DNA inside the cells are cross-linked using formaldehyde to fix the DNA sequences inside the cells. And then this DNA, cross-linked DNA, is digested using restriction endonucleases, creating 5' overhangs. And then these 5' overhangs are filled with biotinylated nucleotides. And then these blunt-ended fragments are ligated, creating chimeric junctions between the nearby sequences. So you can see that these internal biotin tags are present at the center of the ligation junction of these chimeric DNA strands. Then these DNA fragments are shared. And then only the DNA fragments contained in these internal biotin tags pulled out using streptavidin beds. And then the sample is purified such that it is primarily composed of hybrid DNA strands containing informative contacts. And then this DNA is subjected to massively parallel paired Illumina sequencing, finally generating paired and Hi-C reads. And these Hi-C reads can then be mapped back to the draft genome to identify the genome-wide chromatin interactions. So this is an overview of the genome assembly process. So as I mentioned earlier, once we get the hi-fi reads, they are assembled to form contigs at the very first step. So in an ideal scenario, we expect one chromosome to be represented by only one contig, having telomeres at both ends. But if one chromosome is composed of more than one contig, then we need to know which contigs belong to that particular chromosome and in which orientation. So for that purpose, the proximity ligation reads created by Hi-C are useful, where they can be mapped to the draft genome to assign the contigs into chromosomes and also to order and orient them into scaffolds. And this will finally create a fully scaffolded chromosome. So we performed the DNA isolation using cDNA extraction protocol, as mentioned in Furtado 2014 paper. And the PacBio Smart sequencing was performed in 2 smart cells, generating around 2 million hi-fi reads per each smart cell. And the Face Genomics generated around 650 million paired and Hi-C reads. So now let's move on to our first genome, Citrus australis. Citrus australis is a slow-growing, hardy plant, which is naturally found in southeast Queensland. It is commonly known as Australian round lime, dooja, gympie lime, or native lime. These trees are moderately frost-tolerant, and the fruits are globose or subglobose. The seeds of these trees are monoembryonic. So this species is recognized as a resistant species to Huang long bean disease or citrus greening disease, which is one of the most severe diseases causing severe economic losses to citrus industry around the world. So we assembled this genome using hi-fi-SM software. So there are different genome assemblers available to actually assemble the hi-fi reads, such as IPA, hi-fi-SM, Falcon Unzip, and so on. So here we selected the hi-fi-SM software to do that. So hi-fi-SM first performs a haplotype aware error correction to correct the sequencing reads. And then meanwhile, it also keeps the heterozygous alleles intact. So in this image, we can see that this blue color bars are the homozygous alleles, and this yellow and blue color ones, green color ones are the homozygous alleles, and the yellow and blue color ones are the heterozygous alleles. So then it generates a phased assembly graph with the local phasing information using those corrected reads. Here, the alleles coming from only one haplotype is connected in this phased assembly graph. So if we have the global phasing information provided by complementary data, such as Hi-C data, it also can generate haplotype resolved assemblies. So we ran this tool in 3 different modes. In the first mode, only with Hi-Fi reads, it generates a collapsed assembly and 2 haplotypes. So these 2 haplotypes are considered to be partially phased. That means they're not fully resolved. In the second mode, again only with Hi-Fi reads, it generates 2 assemblies. One is the primary, which contains long contigs, but they're not haplotics. And another one is the alternate assembly. It contains haplotics, but they're highly fragmented. In the third mode, which is also known as Hi-C mode, it maps the Hi-Fi reads on a Hi-Fi assembly graph, creating fully haplotype resolved assemblies containing haplotics. So basically, it generates 2 assemblies. One is the collapsed. Collapsed assembly is a complete assembly, and it's kind of like a random mixture of alleles coming from the 2 parents. So the parental alleles switched in the contigs in the collapsed assembly. So these 2 haplotypes are 2 sets of complete assemblies, each representing alleles from only one parent. So with this mode, we observe that it generates assemblies with slightly improved N50s for our Citrus australis genome. So here the N50 is the length of the shortest contig at the 50% of the genome. So most of the time, higher the N50, better the contiguity of the genome, but it's not always true, because sometimes we can have higher N50s for genomes which are misassembled or wrongly assembled. Therefore, it's not the only factor to consider how good a genome is. So we need to look more into deep. So we selected this mode to go for the scaffolding, and the scaffolded assembly is having 31.3 megabases of N50 and 98.8% complete bus scores for the collapsed genome. And the 2 haplotypes are also good in terms of N50s and complete bus scores. Then the assembled genome was, this australis is diploid and heterozygous based on the chem analysis, as you can see on the left side image. On the right side, we have the chromosome scale assembly generated for australis. So in figure A, in x-axis, you can see the chromosome scale assembly generated by Hi-C, and the y-axis shows the contig assembly generated by Hi-Fi SM. So in this image, the blue color diagonal line indicates the 9 lengthiest pseudomolecules, chromosome scale pseudomolecules generated for australis. And the corresponding chromosome numbers were named with the corresponding numbers based on the synteny with the previously published genomes. So here you can see that a yellow dotted line here, it represents it's like medium scale scaffolds which are ranged between 1.5 to 4.5 megabases. They will actually not belong to the 9 chromosomes because they actually contain large clusters of repeats. At the top right corner, we have chloroplast DNA fragments. And figure B shows the individual chromosomes and their corresponding contigs. So you can see that chromosomes 6 and 8 are covered by 2 contigs, and all the other chromosomes are covered by only 1 contig, which is good. And we have 4 pseudomolecules with telomeres at both ends, representing full chromosomes for this genome. But for others, we have 1 telomere. We have telomere at only 1 end. Then this assembled genome was annotated. So the repeats were de novo detected using repeat modeler, and they were masked using repeat masker software. So the repeat analysis revealed that most of the genes are unclassified, like interspersed repeats, where majority of them are unclassified. And among the classified transposable elements, the LTR elements were the predominant type. In addition to these dominant LTR elements, There were some other types of repeats as well in this genome, such as long interspersed nuclear elements, simple repeats, and low complexity repeats, and DNA transposable elements as well. Then we performed the gene prediction by breaker, and it revealed 29,464 number of genes and 32,009 number of transcripts for the collapsed genome. And we also annotated the 2 haplotypes. They are different from each other, and also with respect to the collapsed assembly in terms of the total size and the number of genes as well. So then we performed a synteny analysis between these 2 haplotypes. So the figure on the right side, figure A, shows the synteny blocks between the 2 haplotypes across the 9 chromosomes. So it reveals that most of the gene blocks are in synteny between the 2 haplotypes, but few gene blocks in chromosome 4 and chromosome 7 of haplotype 1 showed synteny with chromosome 8 of haplotype 2, revealing the translocation of these genes between the 2 subgenomes. So figure B shows the synteny of the putative homologous genes of these 2 subgenomes, and it also reveals that most of these genes are in a syntenic arrangement between these 2 haplotypes, with few exceptions, like with few variations in chromosome 4 and 2, where we have inversions. In figure C, it shows the whole genome alignment at the nucleotide level. So it also further confirms a syntenic arrangement of these 2 subgenomes. And we have some structural variations in some chromosomes in these 2 haplotypes. So this basically reveals the differences between these 2 haplotypes, and it's really important to understand these variations to understand the phasing information of this genome. So we performed gene prediction using break, as I mentioned earlier. So this is the pipeline. So in this pipeline, there are 2 gene prediction tools, GenMark and Augustus. So most of these gene prediction tools, including Augustus, they must first be trained on a previously prepared set of genes, and that is known as supervised training. But GenMark has an extraordinary power to be trained by itself, that is known as self-training or unsupervised training. That means it can be trained by itself on a genomic sequence that the GenMark has not previously been trained for. But if we provide the extrinsic data, it also can incorporate this extrinsic data in the breaker pipeline. So this extrinsic data can be either proteins or expression sequence tags or RNA-seq. So here we provide the RNA-seq as the evidence. So we first align the RNA-seq evidence to the genome using high-set aligner, which is a splice-aware aligner. And then that alignment BAM file is directly loaded to the breaker pipeline. So this RNA-seq reads basically being used in this initial training step by GenMark, and also at this last step, last gene prediction step by Augustus. So basically what it does is it first executes self-training of GenMark to produce an initial set of genes, and then those genes produced by GenMark actually breaker selects those having supports in introns in extrinsic data. And those set of genes produced by GenMark is then used as a training set by Augustus to actually produce the final set of genes. Then we perform the functional annotation in omics box. So most of the protein coding genes were given with the protein description based on the sequences in a non-redundant protein sequence database. And the majority of the sequences, majority of the transcript sequences were given at least one geoterm, revealing their molecular function, biological process, and their cellular component. Then we identified some important genes in australis genome. A novel class of relatively stable antimicrobial peptide with 67 amino acids, which is usually known as SAP, has recently been detected from 2 Australian wild limes, glauca and finger lime, and also some other close relatives of citrus, which are recognized as resistant species to HLB disease. The Citrus australis is also recognized as a HLB disease resistant species. So HLB disease actually is now spread across more than 40 countries around the world, including Asia, Africa, and United States of America. It's actually almost all the citrus growing countries in the United States of America. And more than 80% of the Florida industry also has devastated by this disease. So we identified 2 homologous genes encoding antimicrobial peptides. So this first gene, it encodes relatively 2 long peptides. One is 152 and the other one is 192. So they're not 67, but they're relatively long. But the sequence alignment of these peptides that is produced by the resistant cultivars showed that a substantial portion of this 67 amino acids sample version is present as a part of these long peptides. Therefore, we think that it might be possible that these larger precursor proteins produced by this australis may later be modified by proteolytic cleavage to produce the sample versions of the resistant cultivars. Yeah, so I should say that the sequence alignment, according to the sequence alignment, it's not exactly the same. The 67 SAMs are present as a part of these larger precursor proteins with few amino acid substitutions and insertions. Still, they're large and they can be modified later by proteolytic cleavage. And we also identified some other defense-related genes in australis genome. So we identified 76 leucine-rich repeat proteins. Previous studies also have reported enhanced expression levels of leucine-rich repeat receptor kinases in HLB-resistant species with compared to susceptible ones. And we also identified 13 pathogenesis-related proteins which can confer biotic resistance to plants. And we identified 17 guanine nucleotide binding proteins, which have also been identified as highly upregulated genes in HLB-infected fingerling plants. But to further validate these results in australis, we actually need to look at the expression data in australis in response to HLB disease. But actually, currently we cannot do this due to the biosecurity reasons. So most of the commercial cultivars available now, they have no strong tolerance to HLB disease, with few exceptions where some of the varieties have strong tolerance than the others, but majority of them are susceptible to HLB disease. The people have been, because of this reason, people have been using various approaches to actually control the spread of this disease, including the integrating management practices, such as controlling the insect vectors and to use the pathogen-free nursery plants, and also to remove the infected trees as well. But none of these strategies have actually provided a permanent solution to this disease. Therefore, the development of resistant cultivars would be a long-lasting option, or would be a good option. So we can actually use genetic engineering methods to actually enhance the expression of these genes in the variety cultivars of the species. But sometimes the transgenic approaches are not well accepted by the consumers. Therefore, increasing the genetic diversity by citrus breeding approaches to actually introduce the favorable alleles would be another good option. We also were interested in looking at some important genes regulating the citrus acidity. So we identified a few key genes such as pH1, pH5, and their transcription regulators, which are involved in driving the import of citrate into the juice vesicles, and also some other important genes regulating the citric acid degradation inside the cells. And we identified a few key genes governing the synthesis of volatile compound synthesis in australis via 2 main metabolic pathways, NVA and MEP. And we identified many terpene synthase genes as well. So these are the 2 main metabolic pathways which are responsible for the production of terpenes in australis. So here, mevalonate pathway is mainly occurring in cytoplasm, and the MEP or methyl erythritol phosphate pathway is mainly occurring in the plastids. So sesquiterpenes and triterpenes are usually derived from this mevalonate pathway on the left side pathway, and the monoterpenes and diterpenes are usually derived from this MEP pathway. So we identified 37 genes encoding the monoterpenoids, 24 genes responsible for the production of sesquiterpenoids, and 9 genes encoding diterpenoids. So a high diversity of citrus essential oil compounds in citrus can actually provide the resistance of these plants against pests and pathogens. And previous studies also have reported that some citrus plants rich in some monoterpenes and sesquiterpenes actually have antibacterial activities against HLB-causing bacteria HLB-causing bacteria candidate Staphylococcus, and can actually reduce its spread inside the phloem tissue and can provide the resistance for these plants. Therefore, actually, it's really important to understand these volatile compounds, synthetic genes, and their roles in disease resistance. So if you're interested in reading about this research, our paper is now available in horticulture research. You can go and have a look. And then let's move on to our second important genome, Citrus australisica, which is commonly known as finger lime. So finger lime is a unique species among the other members of the Rutaceae family with finger-shaped cylindrical fruits. And also they have a distinct pulp which resembles a carrier. It has a high natural genetic diversity with over 65 varieties in wild, and it is resistant or tolerant to HLB disease and susceptible to other citrus diseases. So we have 5 varieties which are different in skin color and pulp color and taste variations as well. Collate, red finger lime, Rick's red, rainbow, and red champagne. So we try to develop a high quality genome for one of these varieties. We selected here rainbow, and also try to understand the genetic basis of this variation causing pigmentation and volatile compound synthesis and the disease resistance as well. So we performed the same pipeline that we used for australis previously, but with few modifications. And here also, we first started with the high-C integrated mode of the hi-fi SM assembly. And then we aligned the high-C reads to this Hondic assembly using BWA aligner, which is a specialized aligner for high-C. So the proper alignment of high-C reads to the draft genome is critical to get the good results. And this alignment of this high-C reads typically requires specialized or modified aligners because high-C intentionally contains a paired end reads, which is coming from very far away from the same chromosome or maybe between chromosomes. And then this alignment file was subjected to adenoma mapping pipeline to retain only the 5' portion of the chimeric reads and also to remove the PCR duplicates. Then this step actually removes the assembly artifacts, and also it can help to get rid of the misassemblies as well. And then that alignment band file is subjected to SALSA, a scaffolding tool. So here we tried different scaffolding tools like DAS, PinHiC, in addition to SALSA. But for our genome assemblies, SALSA works better than the others. And then this scaffold assembly was aligned against our previously published Genome australis to assign these scaffolds into chromosome level. Citrus australisica finger lime is also diploid and heterozygous based on chem analysis. And on the right side image, we can see the chromosome level genome assembly we generated for finger lime. So in x-axis, it shows the chromosome scale assembly generated for australis. In the y-axis is the chromosome scale assembly generated for finger lime. So in the initial level, same as for australis, finger lime we observed 4 pseudomolecules with telomeres at both ends, so representing 4 chromosomes. But by further aligning these scaffolds against the australis chromosome level assembly, we could further assign some scaffolds into chromosome scale. And now we have 6 pseudomolecules with telomeres at both ends. So in this image, you can see that some of our scaffolds or pseudomolecules have telomeres at both ends, representing the 4 chromosomes. But in some scaffolds, we have telomeres at only one end, here representing as red color, short pieces. But for some scaffolds, we have telomeres at no ends, not at the two ends. So that is because they contain large clusters of ribosomal repeats and satellite repeats at this ends of these chromosomes. So usually Hi-5 reads can span most of the repetitive regions, but here it is unable to assemble these repetitive regions. So as you can see here on this bottom right corner image, most plant genomes constitutes high repetitive regions, such as interspersed repeats, transposable elements, ribosomal RNA gene repeats, and large tandem arrays of satellite repeats. So these repetitive regions, most of these repetitive regions are well conserved among the plant species, but sometimes they are specific to some genera, and also sometimes they are species specific and sometimes chromosome specific as well. So this, because of this variation, people have been actually trying to understand this variation of these repetitive regions and to actually identify the individual chromosomes in plants and also to study about the evolution as well. So anyway, citrus is also characterized as a highly repetitive genomes, mainly present at centromeric, pericentromeric, telomeric, and subtelomeric regions. Thanks to the latest advance in sequencing technologies, most of these repetitive regions can be spanned by high lengths of Hi-5 reads. But still, Hi-5 reads are also unable, as you can see here, unable to assemble some of the scaffolds with these problematic regions into full chromosome level. And we observed another interesting characters in our genomes, citrus genomes. Most of the time, usually, the telomeres are present at the very ends of the chromosomes, but in some citrus genomes, we observe that the telomeres are present at the ends of the chromosome, not as a long continuous sequence, but they are broken at few times and with genes in between them. Usually the telomeres are present as a long continuous sequence with satellite repeats, which are tandemly arranged, but we didn't see that for some of our genomes. And earlier also people have reported that telomere sequences are not always at the very end. They also can be present adjacent to the centromeres and also maybe as a part of the centromere and sometimes interstitial as well. That means between the centromere and the telomere sequences. So for some genomes, we also observed, in our genomes, we also observed telomeric repeats are found in the subtelomeric regions. So it's interesting. And let's talk a little bit about the other Australian wildlife. We have Citrus globe, which is desert lime. It is native to semi-arid regions of Queensland, New South Wales, and South Australia. And this is highly adapted, highly drought-tolerant species, and also adapted to semi-arid regions and saline soils with a deep root system. It shows abiotic stress resistance against salt, boron, and frost, and it also shows biotic stress resistance against VHLB pathogen, nematodes, and it also has been identified as a poor host for viroids. Citrus inodora is commonly known as Russell River lime. It is native to northeast Queensland. It has particularly larger leaves with lemon-shaped fruits, and it is also considered to be a partial resistant species to HLB disease. Citrus gracilis is commonly known as kakaru lime. It is native to eucalypt savanna woodlands of Northern Territory. Citrus garvai is found in Cape York Peninsula. This species also has finger-shaped fruits, which are similar to finger lime. And we also have another unknown citrus species, which is very similar, not very similar, which is similar to garvai. It was also collected from Cape York Peninsula. This species is thought to be a closely related taxon or maybe a subspecies of Citrus garvai. So we have assembled the genomes of all these species. So it's really actually important to characterize all these genomes because they harbor valuable genes which can later be integrated into the commercial cultivars. And let's talk about the 4 domesticated cultivars a little bit then. So we have Mercot. So Mercot is the main Mandarin variety for export in Australia. It's very popular in Asia because of its large size, smooth skin, and high sugar levels. IRN2 is a mutation derived from Mercot, and it has also been an important export variety in Australia. It has many good characteristics compared to Mercot. So Allendale is an old Queensland variety, which is no longer commercially grown in Australia, but it's also been an important variety in Bundaberg breeding programs because of its monoembryonic seed type, large size, and high Brix levels. So Premier is a newly released Mandarin from Bundaberg breeding program, and it is a result from sexual hybridization between Mercot and Allendale, followed by mutation breeding to reduce the number of seeds. So we have assembled the genomes of 4 of these domesticated cultivars into scaffold level. Yeah, with that, I would like to conclude my presentation. So here, for the first time, we have developed high quality, complete, and haplotype resolved chromosome level genome assembly for Citrus australis, and it reveals important genes conferring resistance to diseases and other fruit quality traits as well. So we are trying to assemble the genomes for the other native and for domesticated varieties. We are almost there, but we need to actually develop them into chromosome scale. So this, as I mentioned earlier, these native species are very important because they harbor a lot of valuable genes for biotic and abiotic stresses. And these chromosome scale genome assemblies will be valuable resources for the comparative genomics in the future. And they will also be valuable resources, valuable reference genomes for the other citrus as well. And they will enable us to understand the evolutionary relationships among these species, and finally will facilitate citrus breeding. So these genomes will actually be valuable sources for genomic selection and to perform the genome editing based breeding also, because highly accurate genomes are very important and prerequisite for CRISPR-Cas9 based genome editing to actually select the targets and also to design the guide RNAs as well. Therefore, it's really important to have highly accurate genomes. And in the future, these genomes will provide valuable genomic resources to studying and the assembling of pangenomes, which actually represent a full set of complete set of genes in a plant species or maybe different species, which will further broaden our understanding about the genetic basis of diverse traits, which will be more powerful than relying on a single reference genome. With that, I would like to acknowledge the hot innovation with the co-investments of the University of Queensland and the contributions from the Australian government and via Platforms Australia. And I also would like to thank the Department of Agriculture and Fisheries Bundaberg and Herbalistics Private Limited for providing us with the leaf materials for our study. And I also would like to thank the Australian Genome Research Facility, University of Queensland, for facilitating the PacBio and RNA sequencing, and also for Ramasheti Center for Genomics for providing us with support for high-C sequencing and whole genome short-read sequencing. Then at last but not least, my special thanks also go to my supervisory team, Professor Robert Henry, Dr. Apello Furtado, Malcolm Smith, Dr. Adi Caribbean, and also for my colleagues for providing us with the support and guidance throughout my PhD work. So thank you very much.
Thank you very much, Upuli. We now have time for some questions. And we have the first question in the chat line from Andrew Gearing. Endogenous parararoviruses constitute 1.5% of the genome of Citrus sinensis. Were these genetic elements also present in our native citrus?
What's that, Robert? What's the
there are viruses present in the genome, citrus genome. Did you find any in the native citrus?
No, actually, maybe they're present, but I didn't specifically look at those genes actually. Endogenous pararetroviruses. Yep, I didn't look at those genes.
Something to look at with Andrew, I think.
Yep.
The next question from Phil. Did you find any evidence of subchromosomal translocations between the citrus species?
Did you find any evidence of subchromosomal translocations?
Between the citrus species.
Actually, we have, currently we have actually only assembled one species into chromosome level. So I think it would be really interesting to look at these chromosomal translocations once we assemble all the species, taken as a whole, I think, Robert, because.
Yes, so I think Phil's question will be answered in the near future, but you haven't got quite to that point yet.
Yes, only we have one assembly yet.
Yes. Do we have any other questions? That's all I can find online at the moment. You didn't mention the citrus gracilis genome in your assemblies. What stage are you with that?
Yes, actually I couldn't mention about that. We are waiting for the sequence data of gracilis. We have extracted DNA, RNA, and we have sent the samples for high-C sequencing as well. So we are waiting for all the sequence data. So they will be here soon. So once we get them, we can start assembling that as well. That is the final wild species we are looking for.
Thank you. Do we have any other questions? We have a few moments. We still have quite a few people online. Doesn't seem that we're going to get more questions. The other question I had is what you're planning to do in terms of utilizing the genetic relationships between the domesticated species to understand hybridization and mutagenesis in citrus. Can you explain how you could use those genomes to explore those processes, given that they are related, I believe?
Yes, actually, we have only actually got the high-C data for IRM2, so we have developed the chromosome scale assembly for IRM2. So IRM2 was developed using a gamma irradiation from Mercot. So Mercot, by taking a budwood of Mercot and then applying the gamma irradiation for that budwood, IRM2 has been produced. So actually, basically IRM2 has, like, when we see the evolutionary relationships, phylogenetic relationships between these species, we saw that IRM2 has, like, majority of the nucleotide sequence of IRM2 is, like, basically similar to Mercot. And so we would be interested. It would be interesting to look at these genomes and to understand the genetic variant, the genetics actually caused for the seedless, sorry, not seedless, actually, to reduce the number of seeds causing in this IRM2 genome, because it has reduced number of seeds compared to the Mercot. And also we try to understand, because Premier is a hybrid between this Mercot and Allental genomes varieties. So it would be really important if we can actually develop a Premier as a haplotype result assembly, and to try to understand this phasing information coming from both of these parents. It would be really interesting to look at that as well. So phasing information, yes.
Thank you. Well, if there are no further questions, I think if we can have the next slide.
Yes.
Well, thank you very much for your presentation. And maybe the next slide. So Alice is saying thank you online. Just to remind you that the next seminar will be by Karen McCosker from the Centre for Animal Science on the 23rd of May. And I think that brings us to the end of today's proceedings. So thank you all very much. We'll see you next time. Bye.